Welcome to the PubMed Daily Review for April 16th, 2026. Today we scanned 2,195 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Surgical Interventions and Complications. These papers explore surgical interventions, their complications, and related patient management strategies across diverse medical fields. This cluster covers 512 papers. Representative work includes severe stenosis of major intracranial arteries, an important risk factor for infarction complications after combined revascularization in adult patients with ischemic moyamoya disease. Real-time intraoperative motion following robotic assistance improves efficiency and accuracy in total knee arthroplasty. A retrospective comparative study, and gastric perforation at the extremes of age. A comparative case report of NSAID-associated high-grade dysplasia versus methamphetamine-induced ischemic perforation. Trend 2 of 5, Qualitative Health Research Methods. These papers explore qualitative approaches to understanding health experiences, interventions, and technology adoption. This cluster covers 505 papers. Representative work includes correction. How do patients, medical assistants, and physicians accept and experience tablet-based cognitive testing by medical assistants in general practice? A qualitative study, basis for a qualitative research project, introduction to qualitative research, and basis for a qualitative research project, the research context. Trend 3 of 5, Biomedical and Environmental Innovations. These papers explore novel interventions and predictive models for health and environmental challenges, merging biological and technological advancements. This cluster covers 600 papers. Representative work includes crosstalk of gut-bone muscle, osteosarcopenia in experimental colitis models, healing the unhealable, Wharton's jelly stem cell vesicles as a breakthrough for feline chronic skin ulcers, and areas of patent innovation regarding diabetic foot ulcers, technological prospecting. Trend 4 of 5, Cancer Treatment and Risk Modeling. These papers explore diverse approaches to understanding, predicting, and treating cancer, from novel therapies to risk assessment and diagnostic tools. This cluster covers 290 papers. Representative work includes non-pharmacologic interventions for managing distress, anxiety, and depression for patients with cancer and their family caregivers, a systematic review and meta-analysis, a Phase 1 study of ASDX727 plus talazoparib in patients with triple-negative or hormone-resistant/HER2 negative metastatic breast cancer, and flexible and interpretable modeling of overlapping exposure risks in self-controlled case series analysis. Trend 5 of 5, Disease Management and Immunology. These papers explore how the body, or specific populations, responds to and manages diseases, often through immune system interactions. This cluster covers 288 papers. Representative work includes the role of traditional Chinese medicine in the management of liver disease, targeting gut microbiome, Jiduquisian prescription for systemic lupus erythematosus, a system-level therapeutic strategy for immunological recalibration, and epidemiological characteristics of brucellosis in non-occupational population in Zhejiang Province, China. 2018 to 2024, Epidemic Trend and Infection Analysis. Up next, our first paper out of 5, Multivariate Genetic Analyses of 2.2 million individuals reveal broad and substance-specific pathways of addiction risk by Holly E. Poor and Peter B. Barr from Psychiatry, Robert Wood Johnson Medical School, Rutgers University, New Brunswick, New Jersey, USA. Let's break down its key contributions. Substance use disorders are a major public health concern, and we know they have a significant genetic component. However, these disorders rarely occur in isolation, often co-occurring with other behaviors like impulsivity and ADHD. Traditional genetic studies often look at each disorder separately, but what if there are shared genetic pathways that we're missing? This research explores whether analyzing multiple externalizing traits together can unlock new insights into the genetic underpinnings of addiction risk. To tackle this question, the researchers employed a powerful multivariate genetic analysis technique called genomic SEM. They analyzed genetic data from over 2.2 million individuals, looking simultaneously at substance use disorders and other externalizing traits like ADHD and substance initiation. This approach allowed them to model genetic influences in different ways, either as a single broad externalizing factor or as separate but correlated factors for general externalizing behavior in SUDs. They then used downstream analyses to pinpoint specific genes and biological pathways involved. The study yielded significant findings. By analyzing these traits together, they identified a substantial number of novel genetic loci associated with the broad externalizing factor and, importantly, also found novel loci specifically linked to SUDs. While the number of druggable targets for the broad externalizing factor was high, they also identified specific genes for residual SUDs, suggesting targeted therapeutic avenues. The results strongly suggest that genetic risk for addiction largely operates through pathways shared with other externalizing behaviors, but with distinct substance-specific components. So what does this mean for us? This research strongly supports the idea that understanding addiction risk requires looking beyond individual disorders. By analyzing genetic data in a multivariate way, we can boost our power to discover genes and better understand the complex interplay between addiction and other behaviors. The identification of both shared and substance-specific genetic factors opens doors for more targeted prevention and treatment strategies in the future. This work underscores the power of looking at the bigger genetic picture. Up next, our second paper out of 5, the immunothrombosis pathway linking pulmonary disease to stroke and neurodegeneration. Mechanisms and Therapeutic Implications by Yiping Zhou and Jian Xiao from the First People's Hospital of Jiashan. Jiashan Hospital affiliated to Jiaxing University, Jiaxing, Zhejiang, China. Let's break down its key contributions. We know that lung problems can affect more than just breathing. Increasingly, research shows that conditions like infections, COPD, and even air pollution can trigger a cascade of inflammation and clotting in the body, a process called immunothrombosis. This isn't just confined to the lungs. It can spread and impact blood vessels throughout the body, including those in the brain. This review explores how this lung-brain connection, mediated by immunothrombosis, might lead to serious neurological issues like stroke and long-term cognitive decline. This paper isn't a single experiment, but a comprehensive review that synthesizes evidence from multiple fields. The authors propose a unifying lung-to-brain immunothrombosis framework. They integrate findings from studies on lung diseases, blood clotting, stroke, and neurodegeneration to build a mechanistic hypothesis. The core idea is that inflammation originating in the lungs can trigger a cascade of events involving the immune system and blood coagulation, ultimately affecting the brain's blood vessels and function. The review highlights several key components of this immunothrombosis pathway. These include the activation of endothelial cells, the formation of platelet leukocyte aggregates, the release of neutrophil extracellular traps or nets, complement system activation, and a shutdown of the body's clot-dissolving mechanisms. Various lung conditions, from acute infections like COVID-19 to chronic diseases like COPD and even air pollution, can act as triggers. These processes can then lead to serious consequences in the brain, such as microvascular clots, a leaky blood-brain barrier, and inflammation, ultimately contributing to stroke and cognitive decline. So what does this mean for us? This review strongly suggests that we need to consider lung health as a critical factor in brain health. The immunothrombosis pathway provides a unifying explanation for how lung issues can lead to stroke and cognitive problems. This understanding opens doors for new therapeutic strategies, potentially combining traditional blood thinners with treatments that target specific inflammatory pathways, like those involving NETs or the complement system, to better protect the brain. Up next, our third paper out of 5. Applications and Challenges of CRISPR-Cas Technology for the Detection of Antimicrobial Resistance Genes by Wenting Tang and Meijia Shi from Department of Laboratory Medicine, the second affiliated hospital of Jiaxing University, Jiaxing, Zhejiang, People's Republic of China. Let's break down its key contributions. The rise of antimicrobial resistance is a critical global health threat, leading to millions of deaths each year. Traditional methods for detecting resistance genes, like bacterial culture, can take days or weeks, delaying vital treatment. While PCR is faster, it requires specialized labs and equipment. This highlights a pressing need for rapid, accurate, and accessible diagnostic tools to combat AMR. CRISPR-Cas technology offers a novel approach to detecting antimicrobial resistance genes. It utilizes Cas enzymes like Cas12 and Cas13, guided by specific RNA molecules to precisely target resistance genes. Often, this is combined with rapid isothermal amplification methods. Upon binding the target, the Cas enzyme triggers a cascade of reporter molecule cleavage, generating a detectable signal, paving the way for faster diagnostics. CRISPR-based diagnostics demonstrate impressive performance, delivering results in hours compared to days for traditional culture methods. They achieve sensitivity comparable to PCR, capable of detecting very low copy numbers of resistance genes. Platforms like Sherlock and Detector have been developed, showcasing the technology's potential for rapid and specific AMR gene identification. The clinical significance of CRISPR-Cas for AMR detection is immense, promising faster diagnoses at the point of care. However, challenges such as sample preparation complexity, achieving reliable quantification, and navigating regulatory pathways need to be addressed. Continued research and development are crucial to fully realize the potential of this technology in combating the global threat of antimicrobial resistance. Up next, our fourth paper out of 5, Artificial Intelligence Reshaping the Paradigm of Hematologic Malignancy Diagnosis and Treatment. From Static Assessment to Dynamic Precision Management by Zhu-Jin Li and Weiwei Tian from Bethune Hospital, Shanxi Academy of Medical Sciences, 3rd Hospital of Shanxi Medical University, Tongji Shanxi Hospital, Taiyuan 030032 China. Let's break down its key contributions. Hematologic malignancies, like leukemia and lymphoma, are incredibly diverse. Diagnosing and treating them precisely has always been a challenge, often relying on manual interpretation, which can be subjective. Now, artificial intelligence is stepping in, promising to revolutionize how we manage these diseases, moving us towards more dynamic and personalized care. This paper takes a comprehensive look at how artificial intelligence is being applied across various hematologic malignancies. It reviews AI's role in everything from initial diagnosis and classification to predicting prognosis and guiding treatment decisions. The authors explore advancements from traditional machine learning to sophisticated deep learning and the integration of multiple data types, aiming to provide a roadmap for intelligent clinical pathways. The results are quite impressive. AI models are achieving near-expert performance in tasks like identifying leukemia cells from images. They're also streamlining complex processes like reducing the time needed for minimal residual disease review in flow cytometry. Furthermore, AI is showing promise in integrating multi-gene data for better prognosis prediction and enabling dynamic risk assessments using longitudinal patient data. So what's the big takeaway? AI holds immense promise for making diagnosis and treatment of blood cancers more precise and dynamic. However, we're not quite there yet. Significant hurdles like data sharing, model generalization, and ethical considerations need to be addressed. The path forward involves rigorous validation, standardization, and ensuring AI works collaboratively with clinicians to truly reshape patient care. Up next, our 5th paper out of 5. Deep Visual Proteomics uncovers nociceptor diversity and pain targets by Samperna Chakraborty and Fabian Kasha from Molecular Medicine in the Helmholtz Association, MDC, Molecular Physiology of Somatic Sensation Laboratory, 13125 Berlin, Germany. Let's break down its key contributions. Our senses are incredibly rich, thanks to the diverse types of sensory neurons in our bodies. Scientists have used techniques like single-cell RNA sequencing to map out this diversity at the genetic level. However, the actual proteins within these cells, which are the workhorses that determine their function, are still largely a mystery. This study aims to bridge that gap by using advanced proteomics to explore the protein landscape of these crucial neurons. To tackle this, the researchers employed a sophisticated technique called deep visual proteomics. They started by characterizing mouse sensory neurons using electrophysiology to distinguish between different types, like nociceptors and mechanoreceptors. Then, using laser microdissection, they precisely isolated these individual cell types. Finally, ultra-sensitive mass spectrometry was used to quantify thousands of proteins within each cell, creating a detailed proteomic map. The results were striking. They were able to quantify over 6,000 proteins from each cell type, revealing unique proteomic fingerprints for different sensory neurons. Importantly, they found that these proteomes largely mirrored those found in intact neurons, validating their approach. They also observed that the enzyme B3GNT2 was upregulated in neurons sensitized by nerve growth factor, suggesting it could play a role in pain hypersensitivity. This study demonstrates the power of deep visual proteomics in dissecting cellular complexity. By revealing distinct protein profiles, it It opens doors to discovering novel molecular targets for conditions like chronic pain. This work provides a valuable proteomic resource that significantly enhances our understanding of sensory neuron diversity and the mechanisms underlying pain. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.